I'm going to be talking today about transcription and basically how it's imperative in the protein synthesis that goes on in your body. Alright, so here we are. This is a super simple picture, but basically what I'm using it for is just for the imagery that DNA has the RNA polymerase, which is the arrow, which is an enzyme, and after the enzyme is done with it, it becomes a single strand, and that's basically all that I need for this picture. And then let's see. Alright, so just give me a single second. No, not that one. There we go. Alright, so in the process of initiation, there is what's called an initiator, promoter, a signal that happens, and it comes and latches on to the place where it needs to latch on, and the way it knows that that's the place that it needs to latch on is that it latches onto a data box, essentially. And this box is made up of a series of T's and A's, and it marks the beginning of where the signal needs to start being, or where the DNA needs to start being transcribed. So here, these little rungs, I guess, that you could say on the helix ladder thing, they're nitrogen bases, so there's T's that go with A's and C's that go with G's, and this is the enzyme RNA polymerase, which basically reads, it goes through, first helicase breaks up the DNA into what's called an open complex, which is seen here. So as you can see, this open space is also known as the open complex. And then the RNA polymerase kind of goes on this, what's called a 3' end. And it's just the way it reads. It reads from a 3' to a 5'. And the way we learned it is that it eats 3' and poops out 5', and I'll explain that in detail in a second. So this RNA polymerase is going along this 3', also known as the template strand, and it keeps reading and pooping out until it gets to what's known as the stop sequence, which we learned as the stop codon. It's the same thing. So it starts at the data box and it stops at the stop codon, and it writes. So here, this RNA, it writes from a 5 to a 3, or it poops, whichever way you want to remember it. It poops 5 to a 3, or it writes 5 to a 3, whatever. It reads and writes as it goes. It eats and poops as it goes. Same thing. And the way it knows when to stop is reaching this stop codon, and this part here is still kind of known as the pre-mRNA, and so we'll talk about that now. Alright, so pre-mRNA, handwriting is so good on Paint. Okay, so the difference between pre-mRNA and RNA is because we have eukaryotic cells. We have parts of these, that looks terrible, but we have parts of these RNA strands that we don't necessarily need, or that we don't necessarily want to take out of the nucleus. So we go through a process called RNA splicing. And so what happens essentially is there's all these different basically nitrogen bases, and they're like lining up on this RNA and ready to leave the nucleus and deliver their message, and some of them might not be essential. So say the ones that are pink. So the pink ones aren't essential, but the green ones are. So what's going to happen is so the name of the essential ones are exons, and those are the ones that are ones that we need, and then the introns are the ones that are pink, the ones that we don't need. So what we do is we splice them. So splice kind of sounds like slice, and that's pretty much what it is. They basically slice out what they don't need. So they take that out, and they take that out, and they take that out, take that out, take this out, and they take this out until it's just what they need, until it's just this, let's see, until it's just this line of whatever nitrogen bases that it needs to have. And then before it leaves, so at this point it's called RNA. It's no longer called pre-RNA. So this is now the RNA that will be leaving the nucleus. And so as kind of like a caution, I guess, the RNA strand doesn't just go out. It has what's called a 5 cap on the end. And so that's here, that's the 5 cap, and it's kind of like a battering ram in case something bad happens, like a car bumper, like just in case it loses something. It's like a huge, I guess, rubber helmet on the strand. And then on the end, on the opposite side, there's a sequence of A's. So there's these little nitrogen bases, and they're all adenines. And the point of them is so it's called a poly-A tail basically, and the point of them is like if we lose some in the course of transporting this RNA to whatever ribosome or whatever rough ER it needs to go to, that it's not that big of a deal. So they have kind of like on both sides kind of like a protection. And that's pretty much it. There's this RNA that leaves the nucleus. DNA doesn't leave the nucleus. Once it leaves the nucleus, it goes to either one of two places, which we'll go into the next video, which is translation. And it's coded there in either a ribosome or a or an rough ER, which is endoplasmic reticulum, who knows. I'm really bad at pronunciation. So basically that's it, and there's this A tail, and there's the 5 cap, and hopefully I'm not missing anything. I don't think I am. And yeah, that's it. That's pretty much transcription. It's not too hard. It reads 3 to 5, and it poops 5 to 3, and then it leaves the nucleus. And if it's important to remember, and I'll go over this in the next video, it's important to remember that if it's leaving the cell completely, not just the nucleus, but the entirety of the cell, it's going to go to a ribosome. If it's leaving the entire cell and it's going out of the cell, kind of like going out of a city and using a drawbridge to the next city, that drawbridge is going to be the rough ER. But I'll go into that in depth in the next video. Yeah. Thanks for watching. Bye. My cats are hissing at each other. Bye.